Hello everybody, so today I'm going to be showing you how to use iNaturalist observations to gather species abundance data and how you can refine your sites in a much more specific way. Because one of the issues with iNaturalist is that it's relatively difficult to obtain small sites with the standard user interface. So let's look at the Los Angeles area, for example. So zooming in here, you could use county data, but this is a very large area, and if you want something more specific, this isn't particularly helpful. So you can try and look up, a specific area. We're going to be looking at Eden Canyon, which is a natural area up in the foothills of the San Gabriel Mountains. But if you look it up, and this is common for many sites, it'll take you to sort of a general area that iNaturalist has selected already for you. So this is a popular hiking trail near Eden Canyon Falls. But as you can see here, if I remove this bounding box, we've got tons of information that's here, but there's no simple way for me to draw a box around a particular area. So if we go back to this, let's say we, instead of looking at that area, we want to look down here at Eden Canyon Nature Center. It's a relatively well maintained area. So I'm going to show you how to do that. So what we're going to do here is I'm just going to duplicate this tab. And here you can see we still have the bounding box in the wrong place. It's up there in the mountains, and we want it down here. So what we're going to want to do is go over here to Google Maps. This is pretty easy. And let's go back here for a second. So what you can see here in the URL, if you pay attention here, you've got NE lat, NE long, south SW, so southwest lat, and SW long. So these are your northeast and southwest latitude and longitudes for the corners of your boundary box. So if you go over here back to Google Maps and select your northeast point that you're going to be using as a boundary point, and your southwest point that you're going to be using as a boundary point, you can take your latitude and longitude and just input them into the URL over there. So we're going to go ahead and do that. Just going to copy this, go back over here, northeast latitude, paste, get the longitude, paste that in there, do the same for the southwest corner, and over here, southwest latitude, paste, and southwest longitude, paste. So now go ahead and click enter. It'll refresh your page, and you should hopefully be getting a boundary box in the area that you wanted. So here you can see we got exactly what we needed. We have this rectangle. We're only looking at data that's confined within this area. And this is a pretty good way to look at a site, but there are other ways as well. So another way you could select a site is if perhaps it doesn't fit neatly into a square, like maybe it's not a sectioned off park, you can use a circular selection. And the way you're going to do that is by going up here, and in the URL you're going to delete everything after observations question mark, and you're going to type lat equals, and then leave it blank, and then LNG for longitude equals, you can leave that blank for now, and radius equals, and for now let's leave it at 1. That radius is going to be in kilometers, and it's going to be the radius of your circle. So without pressing enter, we're going to go over here back to Google Maps, and here we're going to select basically a central area where we want the middle of our circle to be. And we're going to find the latitude and longitude. Again, we're just going to copy it, take it over to our other tab. In the latitude, we're going to put in the latitude. The longitude, we're going to put in the longitude. Then go ahead and press enter. Refresh your page. And now you've got a circular boundary with a 1 kilometer radius around this area. So in this particular case, we might want to make it smaller because you can see we're getting a lot of data from the streets here. So you can go in here and change it to maybe 0.5 kilometer radius. You could also change the location again there. So you can get a circle, you can get a custom rectangle, or you can try to just find your area. You know, it might just happen to be populated already by iNaturalist, or you can get, you know, much larger ranges like county-wide data. So once you've got your area selected, for this example, I'm just going to be using the county data. So you can go in here to filters, and here you can see you've got a bunch of different categories and filters that you can use. So I recommend using research grade data, and what that does is it's just observations that have been vetted by professionals in the field, so that you know whatever was observed there, the picture that was taken, you know, it was taken, it's correctly identified, and all the information about it is correctly cited. So for here you have the date range observed. For this, you know, I would recommend maybe you just make it a year's worth of data, so you're getting relatively recent data. You know, obviously this is up to you. Maybe we're just doing a year's worth of data for this example. So let's look at amphibians. You could look at fungi, you know, reptiles, birds, arachnids, whatever. You've got a bunch of taxa here. So once you've got your filter set up and the area set up, you can go ahead and click download, and this will take you to a new page. And this page looks a little complicated, but it's really just the same filter information that you already filled out. So this should already all be filled out. You can see you've got amphibians, research grade, in LA County. This is a preview of some of the information that's there. And here in the columns, this is going to be telling you the data that you're actually downloading, and this will download a very large Excel document. So if you keep all this data in here, you're going to have a really large Excel document that has more information than you probably know what to do with, unless, you know, you're a relatively advanced environmental scientist or biologist, in which case, why are you watching this video? So if you're just taking a simple, taking a simple abundance data for the site, you can get rid of all this, you know, geotagging and identification information, and you can pretty much just include scientific name, and this will just give you a single column that is the scientific name for every observation that was made there. So you're going to go ahead down here and click Create Export, and this will load up, and it'll just take you back up here and give you a download. And here we go, Export Complete, Download. You can go ahead and click that. I've already downloaded this information, so now we're going to head on over to Excel. All right, so here we are in Excel, and this is not the data that I downloaded, but this is an example of what your spreadsheet will look like if you do not change any of those selections that I talked about. So you can see here we've got a very, very large spreadsheet, and really all you need is right here, scientific name. At least this is all you need for simple abundance data. All this other stuff, very specific, potentially very useful for more advanced research, but, you know, beyond the scope of what we need. So basically here you've got what your data should look like. You've got the scientific name. It's just a big old list of all the species that were found, all the observations. So now you're going to go to filter, you're going to go down to advanced, and you're going to be selecting copy to another location. You're going to be copying to the next column over, which is B. You're going to click unique records only, and basically what this does is just compiles each unique instance that a species occurs in that longer list. So you basically just have a list of all the species that were found, that were observed in that location. So now you're going to go in and input this formula, which will be included on the website and in the description. But basically all it's going to be doing is counting how many times that particular species occurs in the longer list of observations. So go ahead and press enter for that and apply it to all of the species, and it should populate. And then now you have abundance data for each species. And now with that, you can use it to make calculations. All right, so now we're in this biodiversity calculator. For this one, we're just calculating Simpson diversity. And this is very straightforward to use. It should sort of automatically populate itself so that if your students aren't necessarily, up to the task, or if it's beyond your assignment to necessarily understand all the math behind it, at least you can understand the concept and get some, empirical data still. So here you're copy and pasting in your data that you got from the Excel document that you downloaded. Here the sum is just the sum of the individuals of each species in that site. The proportion, you're going to populate this down through, for each species. Same with, this is just the proportion squared. And now with that, you should be getting an accurate Simpson diversity value. So obviously this is just a pretty simple example of what you can do with this data. You could also be calculating different diversity indices, such as Shannon diversity. You could go and calculate beta diversity and compare different sites, but this is just a jumping off point and a quick demonstration. What you use this for is up to you, your own research or your own, your own classroom if you're an educator. So yeah, I hope this has been helpful, and thanks for watching.